Good afternoon. The topic of our lecture today is energy metabolism, the common pathway of catabolism. The lecture will cover the concept of metabolism and its main functions, the two sides of metabolism and their relationship, the stages of nutrient catabolism, and in more detail, the common pathway of nutrient catabolism that is typical of all classes of nutrients. We will also discuss the stages and the regulation of this pathway. As you already know, a large number of enzymatic reactions take place in living cells. These reactions occur in a certain sequence and are closely interrelated. The whole set of these reactions is unified by the general concept of metabolism. Metabolism is a set of chemical reactions that occur in the cells of the body from the moment food is ingested until the formation of end products. Metabolism involves many metabolic pathways. The metabolic pathway is a specific sequence of enzymatic reactions in a cell. The intermediate products of the reactions of a metabolic pathway are called metabolites. At each stage of the metabolic pathway, there is a slight chemical change to the metabolites. As a result of these step-by-step transformations, the initial molecule is transformed into the final product of the metabolic pathway. Some metabolites are common to two or more metabolic pathways. Such metabolites located at the intersection of different metabolic pathways are called key or nodal metabolites. Examples of such metabolites include pyruvate, oxaloacetate, and acetyl-CoA. Remember the main functions of metabolism in cells. A. Accumulation of energy released by the splitting of food substances entering the body from the environment in heterotrophs, or by converting captured sunlight energy in autotrophs. B. The transformation of food molecules into structural blocks, which are later used by cells to build macromolecules. C. The formation of proteins, nucleic acids, lipids, polysaccharides, and other cellular components from these structural blocks. D. Synthesis and destruction of biological molecules are necessary for the performance of specific functions by certain cells, such as hormones and mediators. Metabolism consists of two opposing sides, catabolism and anabolism. Catabolism is a phase of metabolism in which complex molecules are broken down into simpler ones. Nutrients that enter the body through food or are produced within the cells are sequentially broken down into compounds such as carbon dioxide, water, and ammonia. Catabolic processes are accompanied by the release of energy. At certain stages of catabolic pathways, a significant amount of this energy is accumulated as a high-energy compound, adenosine triphosphate, ATP. Anabolism is the phase of metabolism during which the biosynthesis of complex molecules, proteins, lipids, and polysaccharides occurs from simple precursors. The processes of biosynthesis proceed with energy consumption. The source of this energy is the breakdown of ATP into ADP and inorganic phosphate. There are metabolic pathways that perform both catabolic and anabolic functions, depending on the needs of the cell. They are called amphibolic, that is, dual. An example of an amphibolic pathway is the Krebs cycle with some auxiliary enzymatic reactions. The processes of catabolism and anabolism occur in the same cell, but their rates are regulated independently. This is because the catabolic pathways and their anabolic counterparts do not completely overlap. The intermediates and individual stages of these pathways may differ. Sometimes the oppositely directed metabolic pathways differ in their intracellular location. At the same time, the catabolic and anabolic phases of metabolism are closely related. A. The energy released in catabolic reactions and accumulated in ATP molecules is consumed in anabolic processes. B. Simple metabolites that are formed in the reactions of catabolism can be used in the biosynthesis of complex metabolites. As already mentioned, catabolism is a set of chemical reactions that convert high molecular compounds into low molecular ones. This is a complex enzymatic process in which it is customary to distinguish three main stages. At the first stage, large molecules are split into their constituent parts. Polysaccharides are converted into monosaccharides. Fats are converted into fatty acids, glycerol, and other components. Proteins are broken down into amino acids. This occurs in the gastrointestinal tract, as well as in lysosomes of cells. The reactions are catalyzed by enzymes belonging to the class of hydrolases. The relative energy output is less than 1% of the total energy released. At the second stage, monomers are transformed into simpler molecules. Monosaccharides, glycerol, and most amino acids are cleaved in the cytosol of cells to the same 3-carbon metabolite, pyruvate. Subsequently, pyruvate, as well as fatty acids and some amino acids, are oxidized in the mitochondria to an acetyl residue associated with coenzyme A, acetyl-CoA. The relative energy output of the second stage of catabolism is about 20%. The released energy can be partially accumulated in the form of ATP. At the third stage, the acetyl group is oxidized in the Krebs cycle to CO2, and reduced forms of coenzymes NADH and FADH2. These coenzymes are oxidized in the respiratory chain to form water, and the released energy is stored in ATP. All these reactions take place in mitochondria. The relative energy input for the third stage is about 80%. There are common and specific pathways of catabolism. The specific pathways of catabolism for substances of a particular class mainly include reactions of the first and second stages, which may differ significantly depending on the class. The third stage, which is the same for all classes of nutrients, is the common pathway of catabolism. In this lecture, we will look at the processes that make up the common pathway of catabolism. The main steps of the common pathway of catabolism are oxidative decarboxylation of pyruvate, tricarboxylic acid cycle or Krebs cycle, and oxidation of reduced coenzyme forms in the respiratory chain, coupled with ATP synthesis. The first two steps occur in the mitochondrial matrix, while the respiratory chain occurs in the inner mitochondrial membrane. Pyruvate undergoes oxidative decarboxylation to acetyl coenzyme A in the mitochondria with the participation of the pyruvate dehydrogenase multi-enzyme complex. The pyruvate dehydrogenase complex consists of three enzymes. 1. Pyruvate decarboxylase coenzyme is thiamine pyrophosphate, TPP, a derivative of vitamin B1. 2. Dihydrolipoyl transacetylase. Its coenzymes are lipoate and coenzyme A, a derivative of pantothenic acid. 3. Dihydrolipoyl dehydrogenase. Its coenzymes are flavin adenine dinucleotide, FAD, a derivative of vitamin B2, and nicotinamide adenine dinucleotide, NAD, a derivative of vitamin PP. Learn the total equation for the reaction catalyzed by the pyruvate dehydrogenase complex. On the left side of the equation, There are pyruvate and two coenzymes, coenzyme A and NAD+. The structure of these coenzymes changes as a result of the reaction. The remaining three coenzymes, TPP, lipoate, FAD, return to their original form at the end of the reaction and are shown above the arrow as a necessary reaction condition. The right side of the equation shows the reaction products: acetyl-CoA, carbon dioxide, and NADH. Acetyl-CoA is then oxidized in the Krebs cycle, and NADH acts as a hydrogen donor for the respiratory chain. The process is aerobic because oxygen is the final electron acceptor for NADH in the respiratory chain. Remember that the formation of acetyl-CoA from pyruvate is an irreversible stage in human and animal cell metabolism. The inability to convert acetyl-CoA into pyruvate explains the inability of the animal body to synthesize glucose from acetyl-CoA and fatty acid oxidation products. The activity of the pyruvate dehydrogenase complex is regulated in various ways: by availability of substrates, by inhibition by reaction products, allosterically, and by covalent modification. Covalent modification of PDH is carried out by phosphorylation and dephosphorylation. The PDH contains two regulatory subunits. One of them, PDH kinase, phosphorylates PDH in some areas through serine residues. As a result of phosphorylation, PDH is inactivated. Another regulatory subunit, phosphatase, dephosphorylates the enzyme, returning it to its active form. When the concentration of ATP increases, PDH is in its dephosphorylated active form. This effect is enhanced in some cells by increasing the concentration of intracellular Ca2+, which activates PDH phosphatase. The products of the pyruvate dehydrogenase reaction, acetyl-CoA and NADH, allosterically activate PDH kinase. Activated kinase phosphorylates and inactivates PDH. Thus, the accumulation of NADH and acetyl-CoA inhibits further conversion of pyruvate into acetyl coenzyme A. Pyruvate allosterically activates the dephosphorylated form of PDH, acting in concert with other substrates, NAD+ and CoA. PDH activation also occurs under the influence of insulin. Insulin increases the concentration of calcium ions in the mitochondria. An increase in the concentration of calcium ions stimulates PDH phosphatase and causes activation of the pyruvate dehydrogenase complex. The Krebs cycle reactions are the third stage of nutrient catabolism and occur in mitochondria. These reactions belong to the common pathway of catabolism and are characteristic of the breakdown of all classes of nutrients, proteins, lipids, and carbohydrates. Remember that the main function of the Krebs cycle is the oxidation of the acetyl group to form two molecules of carbon dioxide, 4 molecules of reduced coenzyme, 3 molecules of NADH, and 1 molecule of FADH2, as well as formation of a molecule of GTP through substrate-level phosphorylation. The cycle occurs in the mitochondrial matrix and includes 8 steps. Let us consider these reactions in sequence, paying special attention to the stages of substrate dehydrogenation and substrate-level phosphorylation. The first step is the condensation of acetyl-CoA with oxaloacetate, which results in the formation of citrate. Therefore, the Krebs cycle is also known as the citrate cycle. In this reaction, the methyl group of the acetyl interacts with the ketone group of oxaloacetate, while the thioether bond is broken. The reaction releases CoA-SH, which can participate in the oxidative decarboxylation of the next pyruvate molecule. The reaction is catalyzed by citrate synthase. It is a regulatory enzyme that is inhibited by high concentrations of NADH, succinyl-CoA, and ATP. The second step is the conversion of citrate to isocitrate via the intermediate formation of cis-aconitate. The citrate formed in the first reaction of the cycle contains a tertiary hydroxyl group and is unable to oxidize under cellular conditions. Under the action of the enzyme aconitase, a water molecule is cleaved off, dehydrated, and then added, hydrated, in a different way. As a result of these transformations, the hydroxyl group moves to a position favorable for its subsequent oxidation. The third step is the dehydrogenation of isocitrate with the simultaneous release of a carbon dioxide molecule, decarboxylation, and the formation of alpha-ketoglutarate. This is the first redox reaction in the Krebs cycle, which results in the formation of NADH. Isocitrate dehydrogenase, which catalyzes the reaction, is a regulatory enzyme that is activated by ADP and inhibited by excess NADH. The fourth step is the oxidative decarboxylation of alpha-ketoglutarate, catalyzed by a multi-enzyme complex, accompanied by the release of another carbon dioxide molecule and formation of a second NADH molecule. Note that this reaction is similar to the pyruvate dehydrogenase reaction. The multi-enzyme inhibitors are reaction products, NADH and succinyl coenzyme A, as well as ATP. Calcium ions act as activators. The fifth step is substrate-level phosphorylation of succinyl-CoA, during which energy released during hydrolysis of thioester bonds is stored as a GTP molecule. Unlike oxidative phosphorylation, the process of substrate-level phosphorylation proceeds without the formation of an electrochemical potential in the mitochondrial membrane. The sixth step is dehydrogenation of succinate to form fumarate, which contains a trans double bond and a FADH2 molecule. Succinate dehydrogenase, an enzyme, is strongly bound to the inner membrane of mitochondria. The seventh step is the hydration of fumarate, which results in the appearance of an easily oxidized hydroxyl group in the molecule of the reaction product. This process is catalyzed by the enzyme fumarase. The eighth stage is dehydrogenation of malate, which leads to the formation of oxaloacetate and a third NADH molecule. The enzyme is malate dehydrogenase. Note that the oxaloacetate formed in the reaction can be reused in a condensation reaction with another acetyl-CoA molecule. Therefore, this metabolic pathway is cyclic. Thus, in one turn of the Krebs cycle, 4 oxidative reactions occur, and 3 NADH molecules and 1 FADH2 molecule are formed. The participation of the Krebs cycle in anabolic processes is due to the fact that some of its metabolites can be used to synthesize monomers, which are used in the building of complex molecules. Thus, oxaloacetate can be converted into the amino acid aspartate and A-ketoglutarate into glutamate. Succinyl-CoA participates in the synthesis of heme, a prosthetic group of hemoglobin, and some other proteins. As a result of the use of Krebs cycle metabolites in anabolic reactions, their amount decreases in mitochondria. Such decrease in the Krebs cycle substrates can be compensated by anaplerotic reactions. The most important of these is the carboxylation reaction of pyruvate into oxaloacetate. Thus, in the reactions of the common pathway of catabolism, oxidation of substrates occurs with formation of reduced coenzymes, NADH and FADH2. The respiratory chain is a sequential chain of enzymes that transfer hydrogen ions and electrons from reduced forms of coenzymes to molecular oxygen, which is the final acceptor of hydrogen. During these reactions, energy is released gradually in small portions and can be accumulated in high-energy bonds in the ATP molecule. The oxidation of NADH and FADH2, coupled with the synthesis of ATP from ADP and phosphate, is called oxidative phosphorylation. The respiratory chain is located in the inner mitochondrial membrane and includes 4 multi-enzyme complexes: NADH coenzyme, Q reductase, succinate coenzyme, Q reductase, and coenzyme QH2 cytochrome C reductase. Cytochrome C oxidase is the last enzyme in this pathway. Complex 1, NADH coenzyme Q reductase, is an integral protein that penetrates the inner mitochondrial membrane. The complex includes electron and proton carriers, flavin mononucleotide, FMN, and iron sulfur centers. Electron donor, NADH. Electron acceptor, coenzyme Q, ubiquinone. The energy released in this reaction is used to transfer 2 protons from the matrix into the intermembrane space. Complex 2, called succinate coenzyme Q reductase, is an integral protein partially immersed in the inner mitochondrial membrane on the matrix side. The complex includes electron and proton carriers, flavin adenine dinucleotide, FAD, and iron sulfur centers. As the first subunit, it contains the enzyme succinate dehydrogenase from the Krebs cycle. The electron donor of the second complex is succinate, and the electron acceptor is ubiquinone. The energy released in this reaction is not enough to transfer 2 protons across the membrane. Coenzyme Q is a fat-soluble vitamin-like substance that can easily diffuse into the hydrophobic phase of the inner mitochondrial membrane. The biological role of coenzyme Q is to transfer electrons through the respiratory chain from flavoproteins, complexes 1 and 2, to cytochrome C, complex 3. Complex 3 is called coenzyme QH2 cytochrome C reductase. It is an integral protein that penetrates the inner mitochondrial membrane, communicating with both the matrix and the intermembrane space. The components of the complex transfer electrons from coenzyme QH2 to cytochrome C. It consists of cytochrome B, cytochrome C1, and iron sulfur centers. Cytochromes are conjugated proteins, hemoproteins that contain an iron ion in an oxidized form. One cytochrome molecule can attach only 1 electron to reduce the iron ion. Therefore, 2 identical cytochrome molecules are needed to transfer 2 electrons. The energy released in this reaction is used to transfer 2 protons from the matrix to the intermembrane space. Cytochrome C is one of the members of the cytochrome family, which contains an iron ion with a variable valence. Cytochrome C is a water-soluble substance that is located on the periphery of the inner mitochondrial membrane in the hydrophilic phase. The biological role of cytochrome C is to transfer electrons in the respiratory chain from complex 3 to complex 4. Complex 4 is called cytochrome C oxidase. It is also an integral protein that penetrates the inner membrane completely and communicates with both the matrix and the intermembrane space. The components of the complex are cytochrome A, cytochrome A3, and copper ions. This complex transfers electrons from cytochrome C to molecular oxygen. The reaction product is water. Due to the energy released in the reactions of the complex, 2 protons are transferred from the mitochondrial matrix to the intermembrane space. The mechanism of ATP synthesis in mitochondria is described by the chemiosmotic hypothesis proposed by Peter Mitchell. According to this hypothesis, the components of the respiratory chain located on the inner mitochondrial membrane capture protons from the mitochondrial matrix during electron transport and transfer them to the intermembrane space. This creates osmotic pressure, which stimulates protons to move back into the matrix and causes ATP synthesis. The transfer of protons from the mitochondrial matrix to the intermembrane space causes the outer surface of the inner membrane to acquire a positive charge and the inner surface to acquire a negative charge. Additionally, a pH gradient is formed with a more acidic pH on the outside. As a result, a transmembrane proton potential is generated with two components, a difference in electrical charge and a pH gradient. The energy of this potential can be used to synthesize one ATP molecule. Protons released into the intermembrane space due to electron transport energy transfer back into the mitochondrial matrix. This process is carried out by the H+ dependent ATP synthase, H+ ATPase. This enzyme is located on the inner mitochondrial membrane. It consists of two parts, a water-soluble catalytic part, F1, and a proton channel immersed in the membrane, F0. The transition of H+ ions from a region with a higher concentration to a region with lower concentration is accompanied by the release of free energy. This energy is used to synthesize one molecule of ATP. The energy accumulated in the form of ATP is used by the body to support a variety of biochemical and physiological processes. Remember the main examples of the use of ATP energy. A. Synthesis of complex chemical compounds from simpler ones, anabolic reactions. B. Performing mechanical work, muscle contraction. C. Generation of transmembrane electrical potentials. D. Transfer of substances through biological membranes against a concentration gradient, active transport. The degree of coupling between oxidation and phosphorylation in mitochondria determines the phosphorylation coefficient, PO. It is equal to the ratio of the number of molecules of inorganic phosphate, H3PO4, converted into ATP to the number of atoms of oxygen consumed, O2. For example, if the hydrogen donor for the respiratory chain is a NADH molecule, then electrons from the donor, NADH, are transferred to the acceptor, oxygen, passing through 3 stages of oxidation and phosphorylation coupling. These stages occur in the first, third, and fourth complexes of the respiratory chain. Thus, 3 ATP molecules, 3 ADP plus, 3 H3PO4, you have 3 ATP, can be formed as much as possible, and 1 atom of oxygen is consumed, 2H plus O, H2O. The value of the ratio PO is 3:1 or 3. If the hydrogen donor is the FADH2 molecule, the electrons in the respiratory chain passing through 2 stages of oxidation and phosphorylation coupling. These stages occur in the third and fourth complexes of the respiratory chain. Thus, 2 ATP molecules can be formed as much as possible, 2 ADP plus 2 H3PO4 each 2 ATP. As in the previous case, 1 atom of oxygen is consumed, 2H plus O each H2O. The value of the PO ratio is 2:1 or 2. The rate of ATP production exactly matches the energy needs of the cell. If the concentration of ATP in the cell decreases and the concentration of ADP increases accordingly, then the processes of tissue respiration and oxidative phosphorylation accelerate. Conversely, if ATP is not used in the cell and its concentration increases, while the concentration of ADP decreases, then oxygen consumption and the rate of oxidative phosphorylation decrease. This dependence of the mitochondrial respiration rate on the concentration of ADP is called respiratory control. Electron transfer in the respiratory chain does not always occur coupled with the phosphorylation of ATP. The state in which oxidation of substrates in the respiratory chain occurs without the formation of ATP is called free oxidation or non-phosphorylating oxidation. The energy released during oxidation is dissipated as heat. Free oxidation under physiological conditions can serve as a mechanism for maintaining body temperature. In pathological conditions, it leads to the development of hypoenergetic states. An example of the involvement of free oxidation in thermoregulation is the metabolism of brown adipose tissue. In humans and some animals, there is a special tissue, brown fat, containing mitochondria, adapted to generate heat. There is a lot of brown fat in newborns, and its amount decreases during subsequent periods of life. In the mitochondria of brown fat, the content of respiratory enzymes is significantly higher than enzymes that phosphorylate ADP. In addition, these mitochondria contain special proteins that uncouple oxidation and phosphorylation, thermogenins. Therefore, free oxidation processes predominate in brown fat cells. Such a thermoregulation mechanism is important for newborns, as they have not yet developed other mechanisms for maintaining body temperature. The uncoupling of oxidation and phosphorylation processes can be caused by the action of a number of substances, both natural and synthetic. The mechanism of action of these substances is that they act as proton carriers across the membrane. Substances that separate oxidation and phosphorylation can be divided into protonophores and ionophores. Protonophores are weak hydrophobic organic acids. They as anions bind protons in the intermembrane space. Then they diffuse through the membrane bypassing the H+ dependent ATPase and dissociate in the matrix to form protons. This group includes, for example, free fatty acids, thyroid hormones, salicylates, dicumarol, and 2,4-dinitrophenol, DNP. The mechanism of action of DNP is shown in the figure. Ionophores are antibiotics, such as valinomycin, nigericin, and gramicidin. They are able to integrate into the biological membrane, forming a channel through which protons and other monovalent cations, such as sodium and potassium ions, can move. As a result, the proton potential is removed and ATP synthesis is disrupted. In aerobic microorganisms, the respiratory chain and phosphorylation enzymes are located in the cell membrane. Therefore, ionophore antibiotics cause the rapid death of microorganisms by stopping energy production and equalizing the ionic gradients between intracellular and extracellular environments.